Hello everyone, my name is Niu Tianxu. Today I would like to present a recent study titled Real-Time Monitoring of Biomolecular Dynamics on Cell Membranes by Quantum Databases Multicolor Electrochemiluminescence. This work was published in Nano Today and include a new method for tracking biomolecule on leaf cells membrane using ECL-based single particle tracking. Among fluorescence material, organic dye are the most common used. They can be excited by laser to emit light at specific wavelengths but suffer from photobleach. Quantum dots are uh, later developing, which can also be excited by laser and emit light at specific wavelengths with narrow emission bandwidth than organic dye. However, laser excitation at high power leaves the high background fluorescence. The overcome photobleaching and high background fluorescence. Electrochemiluminescence excitation was introduced. ECL not only avoids this drawback, but also offer higher brightness and narrow tunable emission bandwidth, making quantum dots an excellent tool for single particle tracking. The picture on the right shows that there we use laser or ECL to excited quantum dot. The wavelength is the same, and different quantum dots give about the same brightness. To enhance ECL signal of quantum dot, co-reactants can be used to provide electron and holes. Among them, oxygen peroxide, an important reactive oxygen species inside cells, serves as a key reactant to boost quantum dot ECL signals. In this study, ECL signal source has two main parts. The quantum dot acts the light emission body. The enzyme glucose oxidase catalyzes glucose in the environment to continuously generate the co-reactant hydroxy peroxide. The functionalized quantum dots are called QGP. By modifying GOX on the quantum dot, the continuous emission process avoids the problem of co-reactant shortage. In this experiment, three quantum dots with emission wavelengths of 525, 580, and 625 nanometers was used. WGA specifically recognized N-acyl glucosamine and salicylic acid molecule on cell membranes. The figures here used quantum dot 525 as an example. Figure A shows TEM image for normal quantum dot and uh, the functionalized quantum dot. Figure B presents the size distribution. Their QGP particles are slightly larger. In figure C, the left half shows the zeta position charge uh, change from negative to the near neutral, reflecting charge in surface chemistry and charge shielding due to the PEG modification. The right half shows the particle size gradually increased, and indicating successful coating with protein and other molecule. In figure D shows the emission spectrum, confirming that QGP-WGA still emit light at the same wavelength as the original quantum dots after functionalization. Figure E and F show that under ECL excitation, the stronger emission intensity is achieved when QGP is combined with both hydrogen peroxide and glucose. Without GOX, the stable hydrogen peroxide production is not stable, so the emission intensity does not significantly increase. In figure G and H show the emission intensity increased as glucose concentration raise up to 4.2 micromolar. Below this concentration, higher glucose results in stronger light emission. The concentration is much lower than typical cell culture media. Their glucose range from 5.6 to 55 millimolar. Therefore, as shown in figure I, the ECL signal remains stable as consistent intensity over a long time in cell culture media. Figure J and K show the voltage range required for excitation and the emission wavelength distribution, respectively. This slide shows the segment of the image experiment. Cells were cultured in dishes with cover slips, then incubate with QGP-WGA at 4 degrees Celsius for 10 minutes. After incubation, the cover slips was taken out and inverted into the specially designed chamber containing wires for imaging. Fluorescence image for A549 cell incubate with unmodified QG showed strong non-specific absorption on the cell membrane. After modification, the non-specific absorption of QGP on the membrane was significantly reduced. QGP-WGA produced stronger ECL signal on the cell membrane. The results confirming that properly modified QGP-WGA can specifically label biomolecule to observe particle target on the cell membranes. Here shows ECL image of QGP-WGA at different potentials. As the potential decreases, the number of individual particle gradual increase, and their intensity becomes stronger. When the potential is further due to below negative 1.1 volt, both the particle number and the intensity remain unchanged, reaching a stable level. Additionally, confocal microscope confirm that the cell membranes stand with the red fluorescence dye DID shows no obvious damage after ECL image at negative 1.1 volt. Picture E shows ECL image at various exposure time. The particle number and intensity increased with longer exposure time. However, beyond 1 second exposure, neither particle count nor intensity increase further. Therefore, subsequent ECL imaging experiments were performed at negative 1.1 volt potential and 1 second exposure time. Three different cell types were used here. A549 as cancer cell, FORT and BHK as non-cancer cell. The hydrogen peroxide levels is higher in cancer cells compared to non-cancer cells. Therefore, when using QD-WGA, cancer cells with more hydrogen peroxide showed a higher particle number. When using QGP-WGA, which is modified with glucose oxidase to continuously and stably produce hydrogen peroxide, the particle number in all three cell types were similar. This demonstrates that QGP can effectively reduce signal variation caused by different cell types and maintain stable ECL signals. Additionally, QD-WGA produced strong ECL signal on the A549 cells membrane initially, but these signals significantly decreased after treatment with vitamin C and hydrogen peroxide inhibitor. In contrast, QGP-WGA maintained high brightness and stability on the cell membrane both before and after the treatment. These results further demonstrate that QGP can resist environmental interference and enable constant and stable ECL imaging on the cell's membrane. Figure A shows the full workflow from image to data analysis. Figure B, C, D compare continuous image of QD-WGA and QGP-WGA in A549 cells. Due to the continuous consumption of hydrogen peroxide, the ECL signal of QD-WGA on the cell membrane decrease and eventually disappear over time. In contrast, QGP-WGA contains glucose oxidase, which continues produce hydrogen peroxide, maintaining stable ECL signals and consistent particle numbers throughout the image period. Using 2D Gaussian fitting method, individual spots in the image are precisely localized. The position of the same particle in each frame are then connected to reconstruct to a movement trajectory on the cell's membrane. This dynamic behavior can be clearly observed in the experimental videos. In this experiment, want to study how molecules move on the cell membrane. The major lipid in manual cell membranes are cholesterol and sphingomyelin. In this study, the PFOD4, which bind with cholesterol, and lysine protein, which bind with sphingomyelin. There are used to mark these two lipids. These markers were linked to quantum dots with different glowing colors to watch how they move on the cell membrane. The results show that WGA signal most appeared where there is a lot of cholesterol or sphingomyelin. And even when WGA moves, these overlaps stay the same, showing that DP and protein interact dynamically on the membrane. Statistical analysis shows that the colocalization between WGA and cholesterol is more extensive than that between WGA and sphingomyelin. The dynamic trajectories clearly demonstrate interaction between WGA and cholesterol as they move together on the cell membrane. The video shows this phenomenon. This page is conclusion. First, advantage of ECL-based single particle tracking. ECL offer low background, high sensitivity, and resists photo bleaching, make it ideal for real-time tracking of biomolecules on cell membranes. Using tunable quantum dots enable multicolor image. Second, design and stability of nanometers. The author combined quantum dot with glucose oxidase and PEG to create nanometers that produce hydrogen peroxide continuously, ensure stable long-term ECL signals for single particle tracking in cell cultures. Third, multicolor tracking and interaction analysis. These systems enable real-time multicolor tracking of biomolecules such as WGA, cholesterol, and uh, sphingomyelin, revealing their dynamic co-localization and interaction on live cell membranes. Thank you very much for your attention.